Okay, and so let's take a look at your proteins. Okay, and so your proteins, they are what they call this macromolecules, no? And so they are organic compounds of high molecular weight. They are macromolecules together with your lipids and your carbohydrates. Okay, and so but then these proteins, they are high molecular weight and they are made up of alpha amino acids. Okay, and so let's take a look at your proteins. Okay, and so your proteins, they are complex molecules. Okay, and so they are complex molecules made up of alpha amino acids. And so with that, if you're going to hydrolyze or break down your proteins, and so you will yield your individual amino acids. And so, and these amino acids, and so when you join them together, you will make a peptide. And the interaction between that individual amino acids combining with another amino acid, and so that is your peptide bonds. Okay, ayan, so your complexity, the complexity of your proteins is related to the combinatorial possibilities of your 20 amino acids. You have actually your 20 amino acids, you have your essential and non-essential. Okay, and so you have there again, and so the diversity or uh, the complexity of your proteins is actually directly related to the combinatorial possibilities of your 20 amino acid monomers. Okay, and so what they call this, your amino acids accordingly, ayan, so are theoretically linked to form your protein. And so that is your via your peptide bonds, no? And so you have there accordingly, and so you have your a total number of combinations of these amino acids, and so can form for at least like a numerous, and so at least trillion, for instance, at least a trillion, no? And so not all this trillion uh possible combinations of your protein sequences is what they call this produced by a living organism. You only have a small fraction, and so you only have a small fraction of these combinations and so of your proteins that is produced in living organisms. Okay, and so let's take a look at those. Let's take a look at why is that. Okay, and so you have there. And so the diversity of proteins is demonstrated accordingly by the complex set of functional and structural properties of your proteins. Okay, and so accordingly, and so you have here your structural features accordingly that make your protein folding a relatively rapid and successful process. And so that is for number one. Okay, and then the presence of your binding sites that are specific for one or a small group of molecules. And then you have here what you call this, there is an appropriate balance of structural flexibility and rigidity so that the function of your protein is maintained. And then you have here your surface structure that is appropriate for a protein's immediate environment. And so let's say, for example, you have your hydrophobic membranes and hydrophilic uh, your hydrophobic and hydrophilic in your membranes. Sorry. And then of course you have your vulnerability and degradation reaction. And so your vulnerability of your proteins to degradation reactions. And so when they become damaged or no longer useful. Okay, and so that is for your proteins still. Okay. Okay, and so accordingly, your proteins can be classified based on the number of amino acids present. Okay, and so let's take a look at that one. Okay, and so they can be classified based on the number of amino acids present. And so your peptides, and so you have for your peptides, you have a few probably for this one. And so under your peptides, you have your dipeptide, your tripeptide, and tetrapeptide, and so on. No, and so you have here Your dipeptide, of course, you have it as it is made up of two amino acids, your tripeptide, and so as the tri denotes, you have at least three amino acids present, no? And then you have your polypeptide accordingly, and so your polypeptide that is made up of 40 to 50 amino acid residues, okay? And so you have it as 40 to 50 amino acid residues for your polypeptide. Okay, and so comparing to the molecular weight of the two of your peptides and your polypeptides is relatively what it calls it has a greater molecular weight. You have it ranges from several thousands to several million daltons. Comparing it to your peptide is less than accordingly 1,000 for that. Okay, and so you have there, and then of course your protein, and so a protein that is made up of greater than 50 amino acids. Okay, and so that is your protein. Again, your protein describes your molecules with more than 50 amino acids. Okay, and so that is for your proteins. Okay, and so accordingly, oligopeptides, if you want to add your oligopeptides, you have at least what you call this uh your oligopeptides, you have a still few, no, ayan, so but then still your oligopeptides, that they are actually smaller than your polypeptides. Okay, let's take a look at your functions of your proteins. Okay, and so your functions, and so first on the list, you have it as your what you call this, your proteins. And so you have your proteins, they provide support and framework to cells and your tissues. Okay, and so you have here listed here, you have your structural proteins like your collagen and elastin. And so other than that, you have your keratin that is present in your hair and nails. And so your keratin also provides structural support, no, and so provides structural support and framework. Again, and so far you have your collagen, and so your collagen, that is actually your major component of your connective tissues, and then your uh elastin, and so your elastin, and so that is a what you call this, a rubber-like protein found in your elastic fibers, and this elastin is found in your elastic fibers. This is found actually, they are found in your blood vessels and skin, and so you have there. And so other than that, you have your fibroin, no? And so your fibroin together with your collagen accordingly. And so your fibroin, this one is from your silkworm or your silkworm protein. Your collagen, and so that is actually your major component of your connective tissues. Okay, and so for your collagen and your fibroin, and so they provide significant mechanical strength. And then you have your movement, and so your movement, and so you have here, and so your proteins, they are involved in your cell movement. Okay, and so under this, under your movement, you have your cytoskeletal proteins like your actin, tubulin, and their probably associated proteins. No, and so they are what you call this, active in cell division, probably endocytosis, exocytosis, and the amoeboid movement of your white blood cells. And then third on the list for this slide, you have your protein function for defense. Okay, and so your proteins function for defense. With that, and so they have many proteins accordingly have a protective functions. Okay, and so you have there in for us, and so for humans, you have your keratin that is found in your skin, and so that is what they call is to provide a protection against a mechanical and chemical injury. And so you have your what you call this, your immunoglobulins, your antibodies, and so produced by your lymphocytes. And then you have your what you call this, acute phase proteins present still in your plasma, your plasma fibrinogen, your fibrinogen. And so these are what you call this, used to prevent blood loss when your blood vessels are destroyed. Okay, and so and actually you have more of proteins that function for defense. Okay, So you have your enzymes, your protein functions. And so with this, your enzyme, you have your catalytic proteins called as your enzymes. Okay. And so your enzymes, they accelerate your, what you call this, biological reactions. Okay. And so under this catalysis as function of your protein, you have your enzymes, they accelerate your biological reactions. Okay. And so you have actually thousands. And so like in the reactions that involved in your digestion, energy capture, and biosynthesis. Okay, and so you have there, and so under this, of course, you have your examples that are included in your in your enzymes. You have your trypsin, your pepsin, your amylase, and the rest of your enzymes. Okay, and so that you know. And then you have your regulation. Okay, and so your proteins regulate also physiological processes. Okay, and so with this, and so you have your hormones for this. Okay, and so binding a hormone molecule, and so accordingly, or growth of factor to receptors in its target cell accordingly changes the cellular function. Okay, and so let's take a look at an example here. You have your the action of your insulin and glucagon, and so both of this, they are peptide hormones, and so that regulate your blood glucose levels. Okay, and so also you have your growth hormone and the rest of your hormones uh, in the rest of your hormones, and so they are involved in regulation, and so regulation of physiological processes. And then you have your proteins, they also function for transport, and so your proteins function for transport, or they facilitate the movement of substances across your cell membranes. And so not only that, facilitate the movement of substances not only across your cell membranes but from an organ to another organ. You have what you call as your carriers. Okay, and so you have your protein again function as carriers of molecules or ions across cell membrane or between cells. And so that is accordingly true. And so examples given here, of course, you have your hemoglobin transports oxygen. And so your sodium potassium ATPase. And so you have there others. You have your uh, glucose transporter, those items. And then your remember that your lipoproteins, they also transport what they call this, they also transport lipids, and so your lipoproteins. And so you have your proteins of functions also for transport. Last on the functions, you have your storage, and so your proteins, and so certain proteins accordingly serve as a reservoir of for your essential nutrients. Okay, and so examples of this, you have your ferritin, and so that is your ferritin accordingly is a protein that is that stores iron. Your of albumin that is actually found in your bird eggs. And so they what they call this they are rich sources of nitrogen during the development of the egg, no. And so you have there, and you have here your proteins, they are function for stress response, no. And so you have here accordingly the capacity of your organisms to survive. Stress is actually mediated by a variety of your proteins. Okay, and so you have here some what you call this proteins can convert variety of toxic materials to less toxic ones. No, and so you have here, and then of course your toxins, and so your proteins, they function for toxins. Okay, and so you have here with this, and so accordingly many uh, organisms produce protein toxins. And so with that, these toxins are actually used in defense and in predation. No, and so examples of this, you have your neurotoxin from your snakes, and so the venom. You have your also your neurotoxin from your what you call this neurotoxin from your scorpions, all those items, and then your apitoxin from the bee sting, and then of course the toxin produced by your bacteria. And so you have the toxin produced by your Clostridium botulinum and the rest of your bacteria. Okay, and so you have here for your what they call this functions of your proteins. Let's take a look at your amino acids. And so, of course, upon hydrolysis of your proteins, you will yield your amino acids. Okay, and so you have here your amino acids, and so these molecules accordingly contain a central carbon atom, or that is referred to as your alpha carbon. And so with that, and so this alpha carbon or your central carbon atom, that is where your amino group and your carboxylate group are attached. And so together with a hydrogen atom. And of course, there is a side chain, your R side chain, your R group, and so are attached also. Okay, and so you have here your R group and or your R side chain, and so varies, and it actually varies and give the amino acid its characteristic. Okay, and so the nature accordingly of the side chain distinguishes your alpha amino acids from each other. Okay, and so the side chain vary in size, in shape, in charge, in acidity, in functional groups present, the hydrogen bonding ability, and the chemical reactivity. Okay, and so that is for your side chain. Okay, and so you have there. Accordingly, your amino acids, they are amphoteric. And so you have your they are amphoteric. And so with that, and so this amphoteric is they can actually behave as either acid or base. Okay, and so accordingly, and so your what you call this, your carboxyl group or the COOH group present in your amino acid, it gives actually the amino acid its acidic property. While and so your amino group, the NH2, gives the amino acid its alkaline or its basic property. Okay, and so at a pH of 7, and so your carboxyl group of an amino acid is its conjugate base. Okay, while the amino group is its conjugate acid. Okay, and so you have there, and so you have here your amino acid, they are amphoteric. Okay, and then of course your amino acids, they are Zwitter ions, and so they possess both positive and negative charges, and so accordingly they possess both positive and negative charges, and so that is called as your Zwitter ions. And so your amino acids, they are also Zwitter ions accordingly, and so accordingly, and so that is at low pH, and so at low pH your uh what you call this, at acidic pH or at low pH, your COOH group accepts a proton and becomes uncharged. And so that the overall charge of the molecule is positive. Okay, at high pH, and so at at alkaline pH, the NH2 group, and so the amino group loses its proton and becomes uncharged. And so giving you the what they call this molecule negative. Again, and so at low pH or at acidic pH, the COOH group accepts a proton and becomes uncharged, and so that the overall charge of the molecule is positive. And then at high pH or at alkaline pH, the NH2 group loses its proton and becomes uncharged. And so giving you the pH is, I mean the charge is negative. Also, and so your amino acids exist in D and L forms, no? And so they also exhibit the optical properties, no? And so you have there. I've said earlier, and so you have your amino acids, and so they are optically active. And so they are optically active. All of actually your amino acids, except your glycine, are optically active. And so with that, your glycine, it is not optically reactive. And so, as what we said, you have the exist in L and D forms. And so the L form, ayan, so that is actually easily hydrolyzed in the body, while the D form is resistant. Okay, and so the L form that is easily hydrolyzed in the body, while the D form is resistant. Okay, ayan, so you have there. And so let's take a look at your what you call this physical properties. Accordingly, your amino acids, they are white and white crystalline, no? And so they are soluble in cold water, except that of your cysteine and your tyrosine. Okay, and so your cysteine, accordingly, and tyrosine are soluble in hot water. Your your amino acids are insoluble in alcohol and ether, except for proline and hydroxyproline. Again, ayan, so your amino acids are insoluble in alcohol and ether, except for your proline and hydroxyproline. And so all amino acids are precipitated off from the solution by alcohol, except proline. And so again, all amino acids are precipitated off from the solution by your alcohol except your proline. And so your proline is not precipitated. Okay, and so and then you have most amino acids are sweet like your uh, glycine, alanine, serine, and proline. And some are tasteless like your leucine, and some are bitter like your arginine. Okay, and so you have there. So these building blocks of proteins can be classified according to their capacity to interact with water. And so that is why you have your polar and your non-polar. And this also amino acids can be classified according to the number of amino and carboxyl group present in the structure. And so you have your what you call as your neutral amino acid, basic amino acid, and your acidic amino acid. Okay, and so you have there your classes. Take a look at your, let's take a look at your polar amino acids. Your polar amino acid, and so the functional group easily interact with water. And so the functional group easily interacts with water. Okay, and so you have there. And so they interact with your water through electrostatic interactions like your hydrogen bonding. Okay, and so examples of your polar amino acids, you have your serine, your threonine, and your tyrosine. And so these ones contain your polar hydroxyl group. And so with this, and so the hydroxyl group present in this serine, threonine, and tyrosine enables them to participate in hydrogen bonding. And so additionally, and so the what they call this, the OH group present, the hydroxyl group present in your serine and threonine are actually points of attachment for your carbohydrates. Okay, and so you have your asparagine and your glutamine, your asparagine and glutamine, they're actually amide derivatives of your acidic amino acids. Okay, and so but then they are polar, and so due to the presence of your amide. And so your amide functional group is highly polar. Okay, and so you have there, and so the amide, your SH, and so the sulfhydryl group of your cysteine is highly reactive accordingly, and what they call this im is an important component in many of your enzymes, and also it binds metals in your proteins. Okay, and so you have here again, and so the what you call this your amide group present in your asparagine and glutamine, and so that is responsible for the polarity of your of these two amino acids. Okay, you have your nonpolar amino acids. Your nonpolar amino acids, and so they contain mostly your hydrocarbon groups. And so your hydrocarbon groups, and so this one do not bear positive or negative charges. And so the what they call this, your nonpolar amino acids, you have it, they are subdivided into two. And so due to the presence of your hydrocarbon R groups, and so you have your aromatic hydrocarbons and then your aliphatic ones. Your aromatic hydrocarbons contain your cyclic structure. And so you have your under this, you have your phenylalanine and tryptophan as the representative amino acid. And in your aliphatic, and so they are non-aromatic hydrocarbons like your methane and cyclohexane. And so a representative of this aliphatic amino acids. You have your glycine, your alanine, valine, leucine, isoleucine, and proline. Okay, and so you have here with this, these are groups, and so you have here, and so your nonpolar amino acids accordingly play an important role in maintaining the three-dimensional structure of proteins, and so due to the fact that they interact poorly with water. Okay, and so you have here just and take note that glycine is has a hydrogen atom in the side chain. And so instead of a hydrocarbon, and this one is your glycine accordingly is slightly hydrophilic. And so accordingly, ayan, so your glycine is slightly hydrophilic due to the presence of a hydrogen atom in the side chain. Okay, and so that is for your non-polar amino. And so you have here your what they call this amino acids, and so that are classified according to the number of your amino and carboxyl groups present in the molecules. And so first on the list, you have your neutral amino acids. Your neutral amino acids, and so they contain in their molecules equal numbers of amino and carboxyl groups. Okay, and so that is why they have a neutral reaction. Okay, and so that is why they have a neutral reaction. Moving on, you have your acidic amino acids. Okay, and so your acidic amino acids, they are monoamino dicarboxylic acids. Okay, and so they are monoamino dicarboxylic acids. Okay, and so with this, ayan, so they have what they call this a uh, negative charge at pH of 7 at physiologic pH. Okay, and so examples, you have two most common, and so representative for this, you have your aspartic acid and your glutamic acid. Okay, and so you have your aspartic acid and your glutamic acid. Okay, and so they have actually a negative, the carboxylate groups are negatively charged at a pH of 7. And then you have your basic amino acids. And so your basic amino acids, and so they are diamino monocarboxylic acids accordingly. And they are alkaline in reaction. Okay. And so you have there your basic amino acids, they bear a positive charge at physiological pH. And so they bear a positive charge at physiological pH. And what you call this, they can therefore form ionic bonds with acidic amino acids. Okay, and so they can form ionic bonds with the acidic amino acids. Okay, and so examples for this basic amino acids, you have your lysine, your histidine, and your arginine. Okay, and so you have there. And so you have here your amino acids. Your amino acids are biologically active. And so you have here your amino acids, your what you call this amino acids and their derivatives, they act as a chemical messengers. And so with this, and so your glycine, your glutamate, and your gamma aminobutyric acid, this GABA here, your gamma aminobutyric acid is a derivative of glutamate. And then you have your serotonin and your melatonin. Your serotonin and melatonin here is a derivative of your tryptophan. And all of this, your glycine, your glutamate, your GABA, your serotonin, and your melatonin, and so they are neurotransmitters. Okay, and so they are neurotransmitters. And then you have your thyroxine. Your thyroxine is a derivative. This is a hormone, sorry, a hormone produced in the thyroid gland. No, and so this thyroxine is a derivative of tyrosine. Okay, and so, and this one, of course, is a hormone. Okay, and so your amino acids also, and so are precursors of what they call this, a variety of nitrogen uh, containing compounds. Okay, and so you have here your examples given here. You have your nitrogenous bases, nitrogenous base components that are present in your nucleotides and nucleic acids. Ayan. So, you have there, and then you have your metabolic intermediates. And so, your metabolic intermediates, you have your amino acids. And so, for example, your arginine, and so derivatives of such, you have your arginine, your citrulline, your ornithine. They are components in your urea cycle. Okay, and so you have here, they are metabolic intermediates, and so you have your arginine, your citrulline, and your ornithine. They are what they call this, uh, uh, compounds, components of your urea cycle. Again, your urea cycle, that is the disposal pathway for your nitrogenous waste. And so these amino acids, and so to form a protein, they need your peptide bonds. And so the bond that is formed between the interaction of one amino acid to another amino acid to form a peptide, that is known as your peptide bonds. Okay, and so your peptide bonds, they are amide linkages formed when your unshared electron pair of alpha amino nitrogen atom of one amino acid attacks the alpha carboxyl atom of another amino acid in a nucleophilic acyl substitution reaction. Okay, ayan, so you have here with this, ayan, so the linked amino acids, if you already have a link, you already formed a hydrogen bond, then you call it as your amino acid residues. Why do you call it as an amino acid residue? Because the peptide bond formation is a dehydration reaction. Okay, and so that is a dehydration reaction. Okay, and so you have there. So let's take a look here. And so you have your peptide bond forms when your alpha carboxyl group of one amino acid reacts with the amino group of another. Ayan, so you have here accordingly. Okay, and so you have your, and of course, what is produced is in water molecule, because remember that as what we said earlier, and so that is a dehydration reaction, and so giving you an amino acid residue and water as the product. Okay, and so let's take a look at your disulfide bridge. And so your disulfide bridge is produced from your cysteine oxidation. And so, but then your disulfide bridge, this one is important in what we call this, the tertiary structure of your protein. And so let's take a look at this. And so your SH group, and so your SH group of your cysteine is highly reactive. Okay, in the most common reaction, and so being it a highly reactive, you have a lot of reactions that can take place with that. And so in the most common reaction is actually the reversible oxidation of the sulfidyl group, and so to form your disulfide bond. Okay, and so your oxidation accordingly of your two molecules of your cysteine, and so forms your cysteine. Okay, and so it forms your cysteine. And so with this, in this oxidation of two molecules, you have a presence of of a disulfide bond. Okay, and so while your disulfide bridge. Okay, and so when actually two cysteine residues form a bond, and so in the peptides or your polypeptides, it is referred to as a disulfide bridge. Okay, and so when two cysteine molecules form a disulfide bond in your peptides or in your polypeptides, and so it is referred to as a disulfide bridge. Okay, and so this bond can occur in a single chain to form a ring, or what we call this, between two separate chains to form an intermolecular bridge. Okay, and so this disulfide bridge helps stabilize polypeptide and proteins. Okay, and so that is for your disulfide bonds produced from the oxidation of your cysteine. So let's take a look at your protein structure. And so under this, you have your primary, your secondary, your tertiary, and your quaternary, no? And so your primary structure, and so this is the specific amino acid sequence. And so with that, of course, and so in your primary, and so the what they call this, they behave as randomly coiled long polypeptides. And so with this, of course, you only have your peptide bonds. There is no folding, And so in this case, okay, and so and then you have your secondary structure. You have here with this, you have here your what you call the secondary structure of your polypeptide consists accordingly of repeating patterns. You have there your folding of your polypeptide chains, and so you have here you're folding here, and so that is a bonding of your carbonyl group of one peptide chain with the amino group of one peptide chain. Okay, and so you have here the what you call this the structures in your secondary structure are stabilized by the localized hydrogen bonding between your carbonyl and the NH group in the polypeptide's backbone. Okay, and so the carbonyl group of one peptide chain with the amino group. Ayan, so that is forming a hydrogen bond. Okay, and so under your secondary structure, you have here your alpha helix and your beta pleated sheet. Okay, and so you have your several units, several repeating patterns in this case. Okay. So let's take a look at your alpha helix. And so your alpha helix, this one is a rigid rod-like structure that forms when a polypeptide chain twists into a right-handed helical for conformation. Okay, and so they are rigid rod-like structure, and they are formed. The polypeptide chain twist when the polypeptide chain twists into right-handed helical conformation. Okay, and so accordingly, hydrogen bonds form between the N to H group of each amino acid and the carbonyl group of the amino acid that is actually four residues away. And so hydrogen bonds form between your NH group of your each amino acid and the carbonyl group of the amino acid that is of four residues away. And so you have actually there. And so giving you actually that a folding pattern. Okay. And so there are actually 3.6, and so there are actually 3.6 amino acid residues per turn of the helix, and so per fold, no, and so per turn, and so in the folds. Okay, and so you have there, and you have what you call as a pitch, and so pitch, and so that is actually the distance between your corresponding points per turn is actually 0.54 nanometers. Okay. And so the uh, R groups, the amino acid R groups extend outward from the helix. Okay, and so you have there for that. Okay, and so okay, and so your not all amino acids can form a helical structure. Okay, and so not all amino acid can form a a helical structure. Okay, and so example, your glycine, your glycine's R group accordingly is so small, and so that the polypeptide chain may be too flexible. Okay, while naman your proline, and so contains a rigid ring that prevents your what you call this nitrogen to carbon bond from rotating. Okay, and so you have there, and so other than that, yeah, your amino acid sequences with large numbers of charged amino acids are actually bulky. They contain a bulky R groups, and they are incompatible with alpha helix structures. Okay, and so that is for your alpha helix. And then you have your beta pleated sheet. And so this beta pleated sheet, and so they are formed when two or more beta strands line up side by side. Okay, and so you have here two or more beta strands line up side by side. And so each beta strand is fully extended. Okay, and so your beta, what you call this, your beta pleated sheets are stabilized by hydrogen bonds that form between your polypeptide backbone. You have here your N2H and carbonyl groups of adjacent chains. Okay, and so they are your beta pleated sheet are either parallel or anti-parallel. Okay, or what you call this? Some what you call this are mixed parallel and anti-parallel. Okay, and so that is for your beta pleated sheet. Look at this structure, and so this one is your uh alpha helix, while on this one is your beta pleated sheet. And so note it is a matted. Okay, you have your tertiary structure. And so with this one, and so your tertiary structure and so forms a unique three-dimensional conformation. Okay, and so it has your three-dimensional shape conformation. Okay, and so you have there. And so this conformation accordingly is a combination of the secondary and tertiary structure present in the peptide chain. Okay, and so your helical arrangement is interrupted with this to allow numerous bonds and additional folding. And so this one is maintained by covalent bonds, and your polypeptide chain is bent or folded to form a compact, tightly folded structure. And so under this, you have your fibrous and your globular proteins. Okay, and so let's take a look at those. So let's take a look first at your interactions that stabilize your tertiary structure. And so with this, and so your hydrophobic structure is first on the list. And so your as the polypeptide folds accordingly, your hydrophobic R groups are brought into close proximity because they are excluded from your water. Again, and so they are hydrophobic. And then, of course, the highly ordered water molecules are released from the interior. And so they are released from the interior, but then, and so uh, they are still they are released from the interior, but then there are still water molecules that remain in the interior, in the core. Okay, and so with this, and so your highly ordered water molecule, and so is released from the interior that will cause a disorder or entropy of water molecules. Okay, and so this entropy is actually a major driving force in protein folding. Okay, and so with that, and so as what we said earlier, you have still water that remain in the core, and so of the molecule, and so this one forms uh the water molecule left in the core of your folded protein forms hydrogen bonds with the polypeptide backbone. Okay, and so that is for your hydrophobic interaction. The list, you have your electrostatic interactions. Okay, and so accordingly, the strongest electrostatic interaction in your proteins occurs between your ionic groups of your opposite charge. No, and so this is also referred as salt bridges, and these salt bridges they are non-covalent bonds, and they occur in regions where water is uh, excluded, no, and so they occur in the regions of protein where water is excluded. And so because of the inner energy required to remove these water molecules from your ionic groups near the surface. Okay, and so you have there. Okay, and so they contribute to interactions between adjacent uh, interactions. And so you have your electrostatic interactions, and then you have your significant. They are significant in the interior of your folded protein and between subunits of protein ligand interactions. Okay, and so you have there for your electrostatic interaction hydrogen bonds. Okay, and so accordingly, a significant number of hydrogen bonds form within a protein's interior and on its surface. And then you have your covalent bonds or your covalent linkages. Okay, and so this one, your covalent bonds are created or formed by chemical reactions that alter a polypeptide structure during or after its synthesis, no? And so you have there, and so the most prominent and on your covalent bond is your covalent disulfide bonds, giving your disulfide bridges. And so these disulfide bridges are found in many extracellular proteins, and so with that they function to protect the protein from adverse changes in pH or salt concentration. And so accordingly, disulfide bridges is absent in intracellular proteins. And so again, your intracellular proteins do not contain disulfide bridge. And then, of course, finally, you have your hydration. And so your hydration, and so you have what you call this, your hydration. In case for this, your structured water is an important stabilizing feature of your protein. Okay, and this one also contributes to the flexibility of the flexibility required for the biological activity of proteins. Okay, and so that is for the tertiary, the interactions in your tertiary structure. Your interactions that maintain the tertiary structure. And so you have your hydrophobic interactions, your disulfide bridge here, your hydrogen bonding, and so you have there. Okay, and so to give you actually to stabilize, to maintain the tertiary structure, to maintain the three-dimensional shape. And then you have your quaternary structure. And so with this, of course, you have your fitting together of your separate coiled and folded structure to form a functional unit. Okay. And so you have your actually in your quaternary structure, you have what you call as your subunit. And so in your subunit, each polypeptide component in a protein complex. And so you have there your subunit that is your polypeptide component in a protein complex. And so they are either identical or different. Okay, and so you have your quaternary, you have several subunits, and so these subunits can either be identical or different. And so you have what you call as your oligomer, and so you have your multi-subunit proteins, and so in which some or all subunits are identical. Okay, and so you have here in your quaternary, and so examples for this, you have your isoenzymes of your enzymes, and then of course your hemoglobin as representative example. And so let's take a look at your common occurrence of your multi-subunit proteins. And so number one, and so the synthesis of separate subunits, and so may be more efficient than substantially increasing the length of a single polypeptide chain. And so accordingly, the synthesis of your separate subunit is more efficient now than increasing the length of a single polypeptide chain. And then the replacement of in what we call the supramolecular complexes like your collagen fibers, replacement of smaller worn out or damaged components can be managed more efficiently. Okay, and so replacement of smaller worn out or damaged components is more effectively managed. And then you have your what we call this the complex interactions help regulate the function of your protein. Again, and so the complex interactions among your multiple subunits present in the single functional protein help regulate the biological function of that protein. Okay, and so that is for your uh, common occurrence of your multi-subunit protein. We are back with your proteins again. And so your proteins can be classified based on your shape. And so you have here, with this, you have your fibrous and your globular. And so there are two major groups. Again, you have your fibrous and globular. And so with your fibrous proteins, and so they are long and they are rod shaped. Okay, and so they are insoluble in water and they are physically tough. Okay, and so with this, a representative example for your fibrous proteins, you have your keratin. And so that is actually your keratin that is found in your skin, your hair, and nails. And so with this, they possess structural and protective function. Okay, and then you have your globular ones, your globular proteins, and so they have your compact and spherical. And so your globular are actually compact spherical molecules. Okay, and so they are usually water soluble. Okay, and so you have your globular proteins. They have a various of functions. They have a dynamic functions. And so examples, you have your enzymes, your immunoglobulins, your hemoglobin, and And so of course your enzymes, and so that is for uh to speed up the reaction. And then your immunoglobulins, they are for what they call this, your immunoglobulins, they are for defense. And then you have your hemoglobin, and so for transport, together with of course your albumin, and then of course your myoglobin. And so you have therefore your classification of your proteins based on the shape. And so you have your fibrous and globular. Okay.